Hello everyone, welcome back to my channel Study Time. In the previous session we were doing the chapter on life process, and in continuation to it, today I'm going to explain the activity 5.1 and activity 5.2. So these two activities are actually the experiments to prove that for the autotrophic nutrition, we know that plants perform photosynthesis, right? And for photosynthesis, few events are very important, like absorption of light energy by chlorophyll. That means the plants should have this pigment chlorophyll, right? And the second is that conversion of light energy to chemical energy and splitting of water molecules into hydrogen and oxygen. So this is the event which is important for photosynthesis for autotrophic nutrition. And then there's reduction of carbon dioxide to carbohydrate, right? So it means that carbon dioxide is very important for autotrophic nutrition for photosynthesis. Then chlorophyll is very important. So today in this topic I'm going to explain activity 5.1, in which we will see that how chlorophyll is important for photosynthesis by performing this experiment. And in the figure 5.2, this is I'm teaching it from your textbook, NCERT textbook for class 10th. So in activity 5.2, I'm going to explain that why carbon dioxide is very important for photosynthesis, right? So these are the essential components of leaf, or during photosynthesis, what all is very important. Now this in activity 5.1, what we have to take a potted plant with variegated leaves, like money plant or croton. We are all well aware of how the money plant leaf looks like. Some of the parts are yellowish in color and some parts are greenish in color in the money plant leaf, as well as in the croton also. So the yellowish part means there is no chlorophyll present in the leaf in those areas, and the green part means the chlorophyll is present in those areas. So what we have to take? We have to take a potted plant with variegated leaves. So these kind of leaves are known as variegated leaves. Some part of the leaf is yellow and some part of the leaf is green in color. And then we have to keep the plant in dark room for 3 days, so that all the starch gets used up. Because generally what happens when the plants are exposed to sunlight, photosynthesis activity keeps on happening, and the glucose is formed, and the extra glucose is stored in the form of starch. So by keeping the plant in dark room for 3 days, what will happen? In the absence of sunlight, photosynthesis will not take place, and the whatever starch is present, it will be used up for keeping the plant alive for different activities in the plant. So we have to make sure that the plant is kept in dark room for 3 days. Now keep the plant in sunlight for about 6 hours, so that again it is exposed to light, and again the photosynthesis starts. Now we have to pluck a leaf from the plant and mark the green areas in it, and trace them on a sheet of paper. Now we have to dip the leaf in boiling water for a few minutes. Now why we will dip the leaf in boiling water? So that it is killed, to kill the leaf, and to kill the cell membrane of the cells of the leaf, and the chlorophyll is also damaged. Then after this, we have to immerse it in a beaker containing alcohol. Now why should we take alcohol? Alcohol is taken so that when we dip the leaf in the alcohol, the chlorophyll is to be removed. To remove the chlorophyll, this leaf is dipped in beaker containing alcohol. Now we have to carefully place the beaker in water bath and heat till the alcohol begins to boil. We cannot boil the alcohol directly, so that's why we have to keep that beaker containing alcohol in another beaker containing water. So that is known as water bath. And so the water becomes hot, and because of that, the alcohol in the other beaker which is placed in that water bath starts to boil. So in this manner, all the chlorophyll is removed. Now what happens to the color of the leaf, and what is the color of the solution? So now what happens? The leaf will become white in color because all the chlorophyll is killed by when we have boiled it in alcohol. And the color of the solution, what happens to the color of the solution? The color of the solution is, it turns to blue. Now why does this happen? Now what we have to do, now dip the leaf in a dilute solution of iodine for a few minutes. Take out the leaf and rinse off the iodine solution. Observe the color of the leaf and compare this with the tracing of the leaf done in the beginning. So we know that this experiment, activity 5.1, by performing this experiment we will prove that when we are putting the leaf in iodine solution, so the starch reacts with iodine solution and the solution will turn blue black in color. So only in those areas of the leaf which had chlorophyll in them, those parts will turn into blackish color. See this one, see. So the green parts contain chlorophyll, and here the starch was present. So starch reacts with iodine and it will turn into blue black color. Rest of the part of the leaf where no chlorophyll was present will remain as such. This one, right? So by performing this experiment we can prove that chlorophyll is very important for photosynthesis, right? I hope it is you all are able to understand it. Now moving on to the next activity. Now this is activity 5.2 from your chapter, and this is page number 83. Okay, so here what we have to do, we know that carbon dioxide is very essential for photosynthesis, so we have to prove it. So this is our setup. So here what we have to do, we have to take two healthy potted plants which are nearly the same size, and then we have to keep them in dark room for 3 days. Just again same thing, that so that no photosynthesis occurs. Now we have to place each plant on separate glass plates and place a watch glass containing potassium hydroxide by side of one of the plant here. So this is one here, the potassium hydroxide is placed here, and then in another setup we will not keep the potassium hydroxide, only in one of the setup. Okay, potassium hydroxide, the function of the potassium hydroxide is to absorb the carbon dioxide. For absorption of carbon dioxide, potassium hydroxide is placed here. Now we'll cover both the plants with separate bell jars. We'll use Vaseline to seal the bottom of the jar to the glass plate so that the setup is airtight. Now we'll keep the plants in sunlight for about 2 hours. We'll again expose the plants to the sunlight. Of course, we have covered it with the bell jar. It is made up of glass, so sunlight will enter, and so we'll expose the plants to sunlight for about 2 hours. Now what we have to do, we have to pluck a leaf from each plant and check for the presence of starch as in the above activity. So what do you think will happen? Do both the leaves show the presence of same amount of starch? Of course no. No. Because why? Here, see, here in one setup we have put the KOH, and here we have not put the KOH. So function of the KOH is to absorb the carbon dioxide. So whatever carbon dioxide was here, it is all absorbed by the KOH. So in the absence of KOH, no photosynthesis will take place when we put it, when we have put these two plants in the sunlight, of course covered by the bell jar. So due to KOH, carbon dioxide was all absorbed. So because of that, no photosynthesis took place here. And in this case, since KOH was not placed, carbon dioxide was there and photosynthesis had taken place. So this last point, do both the leaves show the presence of same amount of starch? No. Answer will be no. And what can you conclude from this activity? We will conclude that carbon dioxide is necessary for photosynthesis. Okay children, so today we did these two activities. Activity 5.1 on page number 82, it is explained. So mostly these questions are asked in your exams, that demonstrate an experiment, or how will you prove by an experiment that carbon dioxide and that chlorophyll is important for photosynthesis. So in that question you will have to explain this activity. Okay, and in the second case, when the question is asked that how can you prove that carbon dioxide is important for photosynthesis, then in that you will explain this experiment. So these two activities are very important part of this chapter in the top under the topic autotrophic nutrition. Right, so I hope you have all able to understand this. So do read the chapter once again, and as I said earlier, in every session I'm telling you that see, this is the book, science book, textbook for class 10. This is of latest edition 2022, and yeah, so October 2022. So this is your prescribed textbook for your schools, in all for CBSE. Okay, so please go through the topic once again, and with this we have completed the portion on autotrophic nutrition. Now tomorrow in my next session I'm going to discuss few questions regarding this, and then we'll move on to heterotrophic nutrition. Right, heterotrophic nutrition. If you have any questions, you can ask in the comment section, and do like, share and subscribe my channel. So I'll be every day I'm going to take the class, and I'm going to discuss this topic. I hope it helps you. Have a good day. Thank you children for attending my class.